Hi everyone, I'm Siddhant from Indian Institute of Technology, Roorkee. So I've been working in Dr. Viva's lab this summer, and I did two projects in this lab, but given the time constraint, I'm going to present just one of them. So yeah, so our lab basically works with tissue engineering. So to give you an overview of the importance of my study, I'll just start with a case study. So this was the case of America's first face transplant. So this is a lady who got shot by his husband in the face, and she lost most of her nose, as you can see here. So this is how she looked on day 0. But what the surgeons did was they took out, they used her inner ear and used that tissue to reproduce her nose. So this is how she looked after the surgery. So this is a technique which is presently used, but this is but this is limited by the availability of autograft. So yeah, so we are looking for a better option for this. So what we are doing is so I'll go with the basic principle of tissue engineering. So in this we took out cells from the patient, we cultured them in the lab or wherever, and we put them into a 3D scaffolds which provide support for the cells to grow inside the cells and outside also. And we use growth factors to promote the growth of those cells. And once we have the tissue, we put it back into the patient's body. And this is how it works. So my lab works in all the 3 areas, but I was interested in the cells. So the question here arises: which cells to use for tissue engineering? So we were interested in mesenchymal stem cells because they have been observed to give rise to differentiate into different lineages like myofibers, neurons, and osteoblasts. So so I'll go for, so where to extract these mesenchymal stem cells from? So the first option is bone marrow. So this is the conventional source of mesenchymal stem cells, but there are a lot of problems with using bone marrow derived stem cells because the surgery which involves extraction of cells from bone marrow is really painful, and there is high donor site morbidity, and the concentration of stem cells is low in bone marrow. So this is the traditional source of mesenchymal stem cells, bone marrow. But we are interested in looking towards adipose tissue for mesenchymal stem cells because the concentration of stem cells in adipose tissue is pretty high. It's higher than in bone marrow, and the tissue itself is really abundant in the body, and it's easily accessible and not painful. So yeah, so we are looking for adipose derived mesenchymal stem cells. So my project was, everything's not visible here, but I guess it's alright. Okay. So my project was to look for the differentiation of mesenchymal stem cells towards myofiber, towards myogenic lineage. So yeah, so this is what my lab previously did. So this is the previous work, just a background. So what they did was they took, they extracted this adipose derived stem cells from a GFP pig and endothelial cells from a GFP pig, and they use C2C12 from mouse. We use pig as a model because that's the closest non-primate model to humans. And they used myoblasts from mouse, and they co-cultured them together in a plate, and they try to look if they can differentiate towards myogenic lineage. But in this study, they differentiated C2C12 are non-differentiated myoblasts, of course. So they put the myogenic differentiation media along with the co-culture. So this is what they already did, and they extracted RNA and all, and did qPCR studies. Yeah, and qPCR studies after different time points, and so this was their result. So what they found out was some of the myotubes were actually green. They expressed GFP, and it shows that the adipose derived mesenchymal stem cells can actually give rise to myotubes. So yeah, and we were so just to make sure that the GFP signal was not because of some leakage through gap junction or something. So they did staining with laminae, porcine specific laminae, and they found that the nucleus was actually red. It's shown with red laminae. So the nuclei were porcine nuclei were actually fused in the myotubes, and they were involved in expression of the genes. So adipose tissues did differentiate toward myogenic lineage, and this is their RT-PCR data, pure RT-PCR data, and they found that myogenin and desmin, which are muscle-specific markers, they were actually expressed higher in the co-culture flask as compared to the control. So my study, so I'll come to my project. So my project was pretty much the same, but this time we tried to do a more practical thing. Like we tried to differentiate C2C12 prior to addition of ADSCs and endothelial cells, right? So because this is more in vivo, like the patient has differentiated myotubes in his body, so we co-cultured them together in this fashion. We used endothelial cells as a control because they are not supposed to differentiate because they are already differentiated. Yeah. So this is my outline. Yeah. So and I did three time points after 12 hours, 24 and 48 hours, and extracted RNA and did qPCR studies. So this is here, and I use desmin and myogenin, which are muscle-specific markers, to as a marker for qPCR. So here are my results. So as you can clearly see that when I used myogenin and desmin, which were porcine-specific, which were porcine-specific, so we can clearly see a rise in the as compared to the control cells over there, which do not show any expression. Very little expression. The co-culture sample actually show a higher level of expression, which was dramatic, like 900 folds. But something interesting happened was endothelial cells, which were not supposed to differentiate towards myogenic lineage, they showed some like 400-fold expression of myogenin and some expression of desmin too. So it may be possible that they redifferentiated and readapted themselves to form myotubes, and that's why we found this expression. So yeah, and we also used myogenin and desmin from murine, from mouse, and so as expected, the control cells like ADSC, my stem cells, which were grown alone, and endothelial cells grown alone did not show any expression because they were from the pig and not from the mouse. And C2C12 show expression because they were from the mouse. But something interesting happened. That was when we co-cultured ADSC, the stem cells, and the C2 differentiated myotubes together, the expression level was higher, and it may be possible because of the prolonged survival of the myotubes, which usually degrades after 3 or 4 days. So it's possible. So here are the conclusion. So the conclusion are the adipose derived mesenchymal stem cells can differentiate toward myogenic lineage when co-cultured with differentiated myotubes. And if we compare this study with our previous study, which was done back in time, so we found that the difference when we cultured them with differentiated myotubes, the expression level was higher than we cultured them when we cultured it with differentiating myoblasts. So this is the conclusion. And what we can do next is we can actually co-culture adipose-derived stem cells with we can actually induce some damage to the myotubes so that it simulates the more patient-like situation where the muscles are actually damaged and all, and then we can co-culture our adipose-derived stem cells with them, and then we can see the effect. And yeah, so this whole study was submitted to International Embryo Transfer Society in Germany for annual conference, and here's the abstract, and here's a quote from Massimo, who helped me in every month. He inspired me. He used to inspire me every time. So I'd like to thank Dr. Peter for accommodating me in his lab. Thank you, Massimo, for inspiring me and helping me. Thank you, everyone, Kelly, Anna, Alisa, Aaron, to be here with me, and Jessica, Stephen, and Rajiv, and Ryan also, because they helped us, to survive. And of course, thank you to University of Illinois and Khorana program. Thank you all.